Staphylococcus aureus, from penicillin sensitive to multi-drug resistant. Antibiotics are medicines that can kill bacteria, and we use it to treat bacterial infections. They are chemical structures that negatively affect some part of the bacteria that is necessary for its survival. The first antibiotic was discovered in 1928 by Alexander Fleming. He noticed that a fungi had grown in the same petri dish as the bacteria Staphylococcus aureus. But there was an empty space between the bacteria and the fungi. Something stopped the bacteria from growing. He discovered that the fungi produced a chemical that killed the bacteria. He called this chemical penicillin. Later, it got made into a medicine as a cure to treat infections caused by the bacteria Staphylococcus aureus. In the 1940s, it was widely used as a new miracle drug against bacterial infections. But the bacteria soon got more and more resistant to the new drug, and by the 1960s, it was said that about 80% of Staphylococcus aureus strains was resistant to penicillin. It had found a way to render the penicillin useless. Penicillin normally works by binding to an important enzyme within the bacteria. This enzyme is important for making a strong cell wall that the bacteria need in order to survive. When penicillin binds here, it stops the enzyme from doing its job, which in turn causes the cell wall to weaken and the bacteria dies. But it turns out that bacteria are very smart one-celled organisms. So Staphylococcus aureus understood that the penicillin made it hard for it to survive and had to figure out a way to destroy it. Eventually, some strains got a new gene. It was a gene that could make a new enzyme called penicillinase, an enzyme that could destroy penicillin. A new bacteria-killing drug was made to combat this problem. The new drug was called methicillin. It has the same effect and binds in the same way as penicillin does, but its chemical structure is a little bit different. The structural difference made it so that the penicillinase enzyme produced by the bacteria would not be able to destroy it. Therefore, this new drug could kill the bacteria. But it did not last long. The bacteria found a way to combat the new drug as well. Both of the antibiotics, penicillin and methicillin, binds to the same enzyme within the bacteria. But if this enzyme is structurally changed, they could no longer bind to it, and the bacteria would survive. So that is exactly what happened to some new strains of Staphylococcus aureus. They got some new genes that made this enzyme a little bit different. It could still do its important job to keep the bacteria alive, but it was different enough to make the antibiotics unable to bind to it. So a new strain of Staphylococcus aureus was discovered, the methicillin-resistant Staphylococcus aureus, or MRSA for short.